Well, hey AP, hope you're doing well when you watch this video. So this is going to be lectures 4, 5, and 6. I'm going to try to make it all at one time just to help you prepare for the test next week. This is all about how DNA goes from a code in a nucleus to a protein. So this is gene expression, okay? So here's our main goals. Understand the code, transcription, translation, and how it's processed. Understand the different parts of RNA and the ribosome and the transporter proteins, okay? So, not the prettiest picture here, right? But our main question for this chapter is, how do antibiotics target bacterial protein synthesis? This is MRSA, methicillin-resistant Staphylococcus aureus. And so, so just keep this in mind. Why do antibiotics work? Okay, what do they do? Okay, and so here is some of the objectives. Starting off, thinking about, how things are made and what's happening, where DNA is. Children with a rare disease called alkaptonuria had parents who were first cousins. Okay, so there's some inbreeding, right? Garrod realized that they must be homozygous for a recessive mutant gene. He isolated homogentisic acid from the children, which accumulated in the blood. So this is normal, this is not. And so this study led to the one gene, one enzyme hypothesis. However, homogenesis as part of a biochemical pathway. So phenylketonuria is another genetic disease that involves this pathway. So let me show you this pathway here. Here's some disorders from phenylketonuria. But here's this pathway, right? So phenylalanine, tyrosine homogentisic acid, okay? And so phenylketonuria, this enzyme doesn't work. Homogentisic acid, this enzyme doesn't work. And so this is a metabolic disorder. Okay, and so the one gene hypothesis, one gene, one enzyme hypothesis, that's what it led to. But studies of proteins made up of multiple polypeptides show that this hypothesis was an oversimplification. So here's the thing. One gene encodes for one polypeptide. Okay, one polypeptide, not just one enzyme, because an enzyme could be made from many different polypeptide chains. So for example, hemoglobin, 4 polypeptide chains, right? So there's 4 different protein chains made to make up hemoglobin, which of course, 2 alphas and 2 betas that help us carry oxygen around, okay? And so this is what led to, the sickle cell disease, which here's the sickle cell disease. So each of these mutant alleles codes for a beta globin polypeptide with an alteration one of its 146 amino acids. So all of these, here's the wild type, these are the amino acids, and here's the differences, variants. You can see some variations don't necessarily cause a ton of possible change in the protein, but nonetheless, this one definitely does. Glutamate to valine, glutamic acid to valine. It would change the structure of the protein, which leads to the change in the function. Okay, so molecular biology is the study of nucleic acids and proteins, often focusing on gene expression. So the mechanisms of gene expression. DNA in genes specify information. The book of you. Okay? Genetic information is expressed into structure and function through protein synthesis. Okay? That protein synthesis, of course, happens at the ribosome. We're talking like the central dogma of biology here. DNA in a gene determines the sequences of nucleotides in an RNA molecule, and then RNA controls the primary structure of a protein. So this is the code of life. So here's your DNA. A binds to T, C binds to G. We've talked about that a lot. And then DNA is transcribed, C comes before L, transcribed into RNA. And then that RNA, let's take off the drawing, that RNA is translated into proteins or amino acid sequences. So codon 1, AGC, would code for serine, GAC would encode for aspartate, and CCC would encode for proline, and that would lead to our polypeptide. Okay, so this is all 3 steps of central dogma: DNA to RNA, DNA to RNA through transcription, and then RNA to protein through translation. Okay, knowing that is crazy important, of course, for moving down the road. So RNA is a polymer of RNA nucleotides. RNA contains a different sugar, it contains ribose, and it contains a different base, uracil. Okay, uracil is definitely And so A binds to U in RNA. 3 different types of RNA. Messenger carries the message out from the gene. Ribosomal RNA makes up a ribosome, and transfer RNA binds to specific amino acids, so they bring the amino acids to the ribosome. And so tRNA recognizes which amino acid should be added next, because it has something called an anticodon on it, which I'll show you a picture here in a second. So you can see right here, this messenger RNA carries a code, rRNA makes up part of the ribosome, and the tRNA, here's the anticodon. So for example, let's write this out. Let's say on the code of RNA it's AUG, which is a start codon, methionine. This would be UAC, the anticodon on tRNA. And so here's a box. I would definitely look at this box. Just memorize that. That would be important. Okay, and so here's the genetic code. Consists of codons, okay? Each of the 20 amino acids is uniquely specified by one or more codons. So this is what it means by the code is redundant, not ambiguous, which means there's more than one way to get amino acid, but there's never like this situation where this encodes for more than one amino acid. That would be ambiguous. Like, which amino acid do I put? They code for both. That's not the case. Every codon has just one specific amino acid it encodes for, okay? UAC, let's just use that. I just made that up. But U, so here's how you read this chart. You start with the first letter U, and you go to A, and then you find the last letter C, and then UAC would encode for tyrosine at the ribosome, okay? And so these are letters, yeah, UACGs. You see function as letters of the genetic alphabet. Codons in the genetic code are all 3 bases long. So again, 64 possible arrangements. Triplets, genetic language only has 64 words. And then here they are, which is built from a code from DNA, which is A binds to T and C binds to G. Of course, not memorizing this at all, giving that to you on the test. And so here's just an example of a code. Cystic fibrosis. This is the protein that makes cystic fibrosis. Over 2,000 mutations could lead to cystic fibrosis. But notice, every single letter, you see this? Every single letter stands for an amino acid. You see right there, that's the entire amino acid sequence of a working cystic fibrosis transmembrane regulator. 2,000 places. 2,000 different amino mutations could cause changes in that, okay? Exceptions. There are exceptions. Retroviruses, like HIV, they actually contain an RNA genome. And then that RNA is converted into DNA. And then that DNA is transcribed and new viruses are produced. Okay, a drug called AZT is a reverse transcriptase inhibitor. Okay, so reverse transcriptase, of course, takes reverse, right? We're going from RNA to DNA in reverse transcriptase. Okay, and then DNA is transcribed back into it, okay? So properties of the genetic code, definitely look over these. Universal, that's an evolutionary term. Redundant, which means 64 codons only for 20 amino acids. Most amino acids are encoded by 2 or more. Unambiguous, which means codons are exclusive. None of the codons right here is super important. None of the codons code for 2 or more amino acids. Contains start and stops. If you want to go back up here, you see 3 stops. Here's the start one, AUG. Always remember AUG, methionine. And then it contains start and stops. Transcription, translation, we talked about that. There's a central dogma. Okay. So here's some questions if you want to go through these. You can ask me questions if you don't understand it. Be good. All right, so this is starting lecture 5. Lecture 5 is how transcription happens, okay? So transcription requires several components. First off, of course, a template, a code, which is DNA. DNA, the book of you, okay? A's, C's, T's, and G's. All right, and so what happens is RNA polymerase. RNA polymerase means builder, right? Polymerase, we're building polymers. We're building RNA from DNA. So RNA polymerases do not need primers, unlike DNA polymerase does. Okay, so here's kind of what it technically looks like. This enzyme from a virus is smaller than most other RNA polymerase, but its active site is similar to those in us. The enzyme is shown in brown, so here's the enzyme. Here's DNA. This is a computer 3D model based upon what's happening, and here's RNA transcript coming out. Okay, so there's 3 parts of transcription: initiation, elongation, and termination. Okay, initiation requires a promoter region. So we just don't read anywhere. We have to look for a region, and that's called a promoter region. This is a control sequence. Understand this definition is important. A control sequence which DNA tells RNA polymerase where to start transcription, where which strand to transcribe. So there's different factors here. So why don't we just always do certain genes, certain things? Because there's factors and transcription factors that bind upstream that basically switch something transcription on or off. Okay? So initiation. RNA polymerase binds to the promoter region. Again, this is all about signaling. Okay? Elongation is when we're actually reading it and building our transcript. So we have building 3 to 5 prime direction. RNA polymerase adds nucleotides to the 3 prime end, so it builds 5 to 3. So it puts the 5 on each time. It's anti-parallel. Okay. And so C binds to G and A binds to U. Not A binds to T. That's only DNA. Okay. So that's elongation. And termination is specified by a specific DNA sequence. They vary among different things. Many proteins are involved, but it's just a sequence that the RNA polymerase dissociates from the reading the gene. Okay. Now, here's our RNA transcript, but it can be changed before it goes on. So this is not a mature RNA transcript because it has to be modified. One of the things that needs to be modified is coding regions. So introns and exons, which ones are kept? Exons. Okay. So introns are non-coding regions, which means they need to be removed to make a functional protein. Okay. So here's what the differences between prokaryotes and eukaryotes look like. But here's an example of what I'm talking about. Here's introns, and these introns need to be removed. Non-coding regions. And so here's the preliminary transcript. We can see here's the actual transcript that we need to make. Okay? And so introns are removed, just segments that need to be removed. Introns interrupt but do not scramble the DNA. Most eukaryotic genes contain introns. Why? Possibly because maybe we need different forms of a protein, right? And so RNA splicing removes introns and splices exons together. If the introns were not removed, would the protein work correctly? And the answer is no. The protein would have more amino acids, right, in its structure, thereby creating a different 3D look and possible function. Okay? So you need to have the introns removed. And a spliceosome does that. Okay? Introns are removed. Exons are spliced together. But yeah, the reason I put a star here is because alternative splicing may allow different versions of mRNA. So we might be able to make a lot of different mRNA versions from the same gene sequence, which allows for us to kind of adapt to what the environment's doing. What do we need? What signals are we getting? Introns, why do we even have them? Possible functions. Exons might combine in various combinations to give different proteins depending upon the environment. But introns might regulate gene expression. Maybe we don't need something we're making. Introns may encourage crossing over during meiosis. Okay, it's a really fascinating time in genetics. I mean, it's just pretty crazy to think about. So here's what the spliceosome looks like. You can see 2 different proteins here binding. Here's them binding again together. Cut one side, cut another side, match it up. Just an example of here in the disease beta thalassemia, a mutation at an intron sequence in the beta globin gene prevents the pre-RNA from being spliced correctly. So what happens? a non-functional RNA is produced, which of course leads to severe anemia. Again, we're talking about beta globin and hemoglobin. Mutations are used to elucidate cause and effect relationships. The other pre-mRNA modifications are as follows. You need a 5' cap, which is added to the 5' end. That's pretty straightforward. And so what happens is this is just protection. It prevents digestion from enzymes that are recycling stuff. And so it's a modified guanine nucleotide. So it helps a ribosome determine where to attach when translation begins. The poly A tail helps stability. It helps get the mRNA out of the nucleus. Okay, those are two other. And so this is kind of what it looks like. Cap, poly A tail, protection, helping it get out of the nucleus. And this is just what it looks like again. Here's the cap, lots of guanines together. And then here is the poly A tail, 50 to 250. Again, these are spliced out. These are removed. Of course, the protein coding sequence is right here. So it would stop, this protein would stop moving right there, and that's just another picture. Here's some questions that would be good to go through. All right, guys, thanks for hanging in there so long. So here is lecture 6, and here's the final lecture for the chapter. And this is all about translation. Again, lots of great Amoeba Sisters, lots of different videos out there to watch about this. Translation of the nucleotide sequence into, we're going from RNA to amino acids, right? Prokaryotes, they're coupled, transcription and translation. Eukaryotes, it separates these different processes. And so you can see right here, codons, 3-letter words called codons, specify one amino acid. They figured this out by actually just making tons of U's and tons of C's, and they saw, which amino acids were put together in 1960 something, I believe. And so there's start codons, AUG, and then there's stop codons. For most amino acids, there's more than one codon, so the genetic code is redundant but not ambiguous. That's a very important phrase. Redundant, which means multiple codons can code for the same amino acid, but not ambiguous. No codon codes for more than one amino acid. Talked about that a couple times. Definitely important that you kind of think that through. Okay, here's the codon chart again. This one's probably a little nicer to look at. You can see right there, there's a start. Here's the stop codons. Tyrosine has a few. Proline has 4. What's the one that has like 6? Arginine, 6. So there's a lot of different redundancy examples. Universal, genetic code is ancient. So unifies life. Indicates that all life came from a common ancestor. Of course, that's an evolutionary term. Genesis 1 teaches that God created the heavens and the earth. And it also is the basic for genetic engineering, which is basically like having bacteria make proteins that are found in humans. People don't die from diabetes. Why? Because of genetic engineering, which is crazy kind of think about. Different mutations can happen. I made the lecture 3 video deals with mutations. But point mutations also confirm the genetic code. So point mutations are compared with the amino acid sequence. Silent mutations occur because of redundancy. So for example, let's say a G becomes a U. Well, it doesn't matter because it's still going to give the amino acid proline. So you can see right here, proline, and then still proline, right? Missense mutations result in a change in the amino acid sequence. Could be good, could be bad, but GUU is valine, which again is a big molecule, so that would be a problem. And that's why sickle cell. You can see right here, glutamic acid, normal, good. Valine changes the complete structure of the protein. Sickle cell, one letter out of hundreds and hundreds changes that. Okay, nonsense mutations result in a premature stop codon. This is definitely usually not functional because, you can see right here, you have one amino acid. That's not protein's not even made there. And then frame shift are usually pretty deadly or dangerous because it's deleted, insertion or deletion. So you can see right here, we were supposed to make CGA, but a T's been added, and that completely changes all the rest, okay, which is a problem. So here is tRNA, 2-dimensional, 3-dimensional. Simplify, there's the anticodon, okay? Two events must occur to build a protein, okay? tRNA must read mRNA correctly, and they must deliver the correct amino acid, okay? Amino acids are achieved from eating. Essentially, you have to eat, okay? Have to eat them. Got to get them inside your body somehow. And then here's just an example of an amino acid protein file from Sun Warrior, egg, and whey protein. Sun Warrior is like a plant-based protein. If you take protein powder or anything like that, you can see the differences here. Everything has an amino acid profile because protein, you're trying to build muscle. And so the structure of tRNAs facilitates their three functions. Number one, they bind to a specific amino acid becoming charged. Okay, formally it's a function. Bind to mRNA at the anticodon, that's complementary base pairing. A binds to U, C binds to G. And then, so let's write that answer right here. So 5' CGG 3' would be 3' GCC 5'. Okay? Non-covalently, of course, because we want to dissociate. And there's just tRNAs again. And so here is normal pairing. Sometimes there is an interesting thing that AP Classroom wants you to know as far as the wobble effect. So specificity for the bases is not always observed. Sometimes it's like, oh, the first two's good, let's rock and roll. Okay, so codons for alanine are recognized by the same tRNA. So the wobble allows cells to produce fewer tRNA species, conservation of energy here, but does not allow the genetic code to be ambiguous because they're all encoded for alanine. And so here's just tRNA, and enzyme has to charge them, build them together. There you go. It takes some ATP to do that. We're building something. And then charge RNA there. Okay. Translation, where does it occur? It occurs at ribosomes. So all prokaryotic and eukaryotic ribosomes consist of one large and one small subunit. Here's a large, here's a small, three different sites, EPA. Here's just an example there. Okay. And so A, it's a rival site with the anticodon. P is where the site tRNA adds its amino acid, and E is where it exits. Okay, so here's a small subunit, large subunit. Start codon always starts with start codon. It might be spliced out eventually or changed out or modified at the Golgi or something, but it always starts with methionine. And there's a couple of different different processes here. Number one, initiation. Initiation, the small subunit binds to its recognition sequence on mRNA. Methionine then binds to the AUG start codon. That's initiation. Looking for that start codon. Elongation. So it enters the A site, remember it's EPA, and the large subunit catalyzes two reactions. Breaks the bond, forms the peptide bond. There you go. Arrives, bond, exits. Okay? So the component of this activity is an rRNA, so the catalyst is an example of a ribozyme. So this is part protein, part RNA. And what's happening? This is the bond that's happening. Water is being removed. So this is a condensation reaction. Dehydration, we're removing water, there's our peptide bond, amino acids are joined together, and the amino acid sequence will be the primary sequence, leading to how a protein folds. Okay, first tRNA is released, can become charged again if the situation calls for it. Elongation, EPA, and then translation ends when a stop codon enters, there's a release factor, and it releases. Okay. Several ribosomes can simultaneously translate a single mRNA molecule. Yeah, some mRNA molecules are crazy long. We're talking like thousands upon thousands of amino acids, so you need to save some time with that. And so here's some questions again for you to practice with. And thank you so much for watching. God bless.